Good morning, everybody. Is anyone listening to me? Can you listen to me? Okay. Today we have a great privilege having Amalia Gastaldelli as a guest speaker. And there is an old Italian saying that a good day starts with a good morning. And since this is the first yellow webinar of the year, I'm sure that this will be the best ambassador for a successful 2025 series. There is no need actually to present Amalia. Amalia is a worldwide well-known researcher dealing with the pathophysiology of a very hot topic today in hepatology, which is the fatty liver, NAFLD, MASLD, and as you like to call it. And the great advantage is to link this with also the diabetic alteration, which actually plays a role in the overall clinical picture. Amalia is a professor at the University of at the CNR, which is the research institution of the National Research Council in Pisa, and also associate adjunct professor of medicine and diabetes nutrition in the University of Texas at San Antonio. She's most important coordinator and the president of the European MASLD Study Group affiliated with the ASD, and she's also president of the European Group for the Study of Insulin Resistance. She's a shining star, and we are very privileged to have her here with us today. And please, Amalia, the podium is yours.
Thank you very much, Claudio and Natalia, for this invitation. This is a great privilege for me today to present some of the work we have done in the past year. And as a title, I chose Metabolic Alteration in MASLD, Lipids, but also glucose. And I will start with my disclosure. And so I like to start with this is the pathophysiology of diabetes and MASLD, because they all share a common pathway. And we know that a positive energy balance, especially with an excess of fat and refined sugar, is associated not only with obesity, but also with all the other metabolic diseases, and also with alteration in metabolic fluxes. And this is what I do in my research. We measure in vivo in humans and also in animal models these metabolic fluxes to understand what is really driving the deposition of fat, the alteration in fat production, and also for diabetes, the changes in glucose metabolism. So if you see here, this is, let me just put the laser pointer so will be easier. So that when there is an excess energy substrate, the first is to try to store it in the adipose tissue, but sometimes the adipose tissue cannot store the entire excess energy, and so there is a lipid spillover. The adipose tissue becomes inflamed and insulin resistant, and this lipid spillover is also going to other tissues that are the main one is, of course, the liver, but you can find some lipid deposition also as ectopic fat in other organs, mainly the muscle and the pancreas. So with that, you will have an alteration in both muscle function and insulin resistance, but also in the secretion of important pancreatic hormone like insulin and then glucagon, that together with an excess fatty acid that spill over from the adipose tissue, they go directly to the liver, where they are involved in not only the accumulation as lipid drops, as the is full of triglyceride, but also of the other lipotoxic lipids. So there is a close relationship between MASLD, MASH, and type 2 diabetes. That is not only the insulin resistance, but also the excess flux. In MASLD, you have an excess of lipids and lipotoxicity. In diabetes, there is hyperglycemia and glucotoxicity, but the two are really interrelated. And of course, the adipose tissue is making a big job, because as I said, when it cannot expand and get all these excess lipids is also playing an important role. So this alteration in biology is associated also with insulin resistance in other organs, not only the adipose tissue, but also in the liver and the muscle. And this inflamed adipose tissue is usually have impaired oxygen metabolism, is becoming also fibrogenic, release pro-inflammatory adipokines, the increased expression of pro-inflammatory proteins, increasing the excretion of metabolic deleterious exosomes, has a decreased catabolism of branched-chain amino acid, decrease adiponectin production, and increase release of other adipokines that are pro-inflammatory, and of course, an increased lipolytic activity. So there is, as I was saying before, important to study insulin resistance and increase metabolic fluxes in these metabolic diseases. And we do this by using the substrate that are labeled with stable isotopes. You see here that to study lipolysis, we usually use labeled glycerol with deuterium or palmitate with deuterium of carbon-13. These are all stable isotopes and non-radioactive. For gluconeogenesis and de novo lipogenesis, we use the deuterated water. For glucose metabolism, we use glucose labeled with deuterium or C13s, and also not only to look at the liver, but also to look at the muscle. So with for the glucose uptake, and also amino acid to look at muscle catabolism. Of course, today there is no time to look at all these fluxes, so we'll just focus on some of them. And of course, especially, these fluxes are important to measure the degree of insulin resistance in each tissue. As I said, insulin resistance can occur not only in the muscle, that was the most studied, but also in the adipose tissue and in the liver. The adipose tissue insulin resistance is associated with increased lipolysis that goes to the liver, hepatic insulin resistance with increased glucose production and the VLDL secretion, and in the muscle that is associated with insufficient glucose disposal. Excess lipids are stored in the tissues as lipid droplets and released by lipolysis. And this is important because knowing how these are the metabolism. Maybe you can switch off his. MASLD insulin-resistant lipid fluxes are important for MASLD, and I will start with the lipid metabolism to show you some of the results. So by infusing glycerol that is used to look at lipolysis, we measure together with Professor Bugianesi in Torino that subjects with MASLD have an increased release of free fatty acid. You see here the profile of plasma free fatty acid during insulin infusion. So insulin is able to suppress free fatty acid, but to a less extent in subject with MASLD. Subject with MASLD also have an increased lipolysis. But when we look at the oxidation, we were thinking to find a decreased lipid oxidation associated with fat deposition in the liver, but actually these subjects have an increased total lipid oxidation and increased hepatic lipid oxidation measured by the levels of beta-hydroxybutyrate that is the product of beta oxidation. Showing that there is really an association between the excess lipids to the liver. The liver tries to detoxify the excess lipids by increasing oxidation, but is not sufficient, and so is much higher than in the controls. So increased lipolysis, but not decreased fat oxidation in MASLD, and this is related, to adipose tissue insulin resistance. So how to measure this adipose tissue insulin resistance? Of course, if you don't have tracer, you can use other indexes. The one that we have developed and that has been widely studied, widely used not only by us, but also by many other groups, is the product by free fatty acid times insulin, because as insulin concentration are higher, you are expecting a decrease in free fatty acids. But when you have insulin resistance, you need more insulin to reach the same levels of free fatty acid. And this curve can be described by this product. So what we have shown, you see here the dates many years ago, is that the adipose tissue insulin resistance index is much higher in MASH than in controls. That is almost 4 times higher, but also it's increasing with the degree of diabetes. So a subject becomes obese, glucose intolerant, or diabetic. They have an increase in adipose tissue insulin resistance. And also, this insulin resistance due to the impaired suppression is called also inflexibility. And you see here that it's associated with the amount of fat in the liver. And this is the same for subject with or without diabetes. This is a study done by the group in Maastricht. Subject with NAFL, independent of diabetes, they have a much higher adipose tissue insulin resistance, and also is associated with the degree of steatosis. So here the degree of steatosis, quantile of hepatic triglyceride content, you see the suppression of free fatty acids decreasing linearly with the amount of fat in the liver. But also is associated with severity of liver fibrosis. So it looks like as more the liver becomes stiffness, or as measured by liver biopsy, the degree of fibrosis, there is a higher hepatic adipose tissue insulin resistance, because these free fatty acids that arrive to the liver to gravitate with insulin are also associated with they are prone to increase the inflammation in the liver and the generation of collagen and, of course, liver fibrosis. So which is the impact then of de novo lipogenesis in the liver? De novo lipogenesis is the de novo synthesis of lipids, starting from a precursor that are usually is acetyl-CoA that is available from refined sugars, mainly fructose. So remember that sucrose that contains one mole of glucose and one of sucrose and one of fructose is one of the most toxic nutrient compounds for the liver, and together with insulin that promotes the synthesis of free fatty acid and activate FASN, there is a synthesis of saturated fatty acid. The first one is palmitate, but also myristate, and stearate for elongation. And these saturated fatty acids are very toxic for the cells, and so they need to be used up to make other lipids or desaturated. So the amount of DNL is also associated with increased saturated fatty acids. And so it's also important because de novo lipogenesis is associated with MASLD, presence of MASLD more than obesity per se. You see here one of the study by group of Dr. Sam Klein in St. Louis. Just having obesity is not associated with increased DNL, but obesity and NAFLD have a very high contribution of DNL to triglyceride palmitate, and you see here that the contribution of DNL is increasing with the hepatic fat content, but also there is a strong relationship between insulin resistance and hepatic and muscle insulin resistance and DNL. Also, the diet composition influence hepatic DNL and adipose tissue lipolysis. This is a study our group has done together with Professor Iki Arvinen. So looking at excess 1,000 calories for 3 weeks in subjects that were fed with excess saturated fat or unsaturated fat or simple sugar, we found that the increase of de novo lipogenesis was much higher in the group that have excess simple sugars with the carb, and followed by the one that had excess saturated fatty acid. But the change in intrahepatic TG was actually much larger in those with saturated, with a diet rich in saturated fatty acids, that also, for some reason still unknown, they also have an increase on peripheral lipolysis, and so the peripheral lipolysis and the increase in fatty acids from the diet really drove the hepatic triglyceride accumulation. Again, lipolysis is the main driver, but we need to think also about how the liver wants to take out the lipids. So it's usually done by VLDL secretion. So one of the question was, is VLDL secretion decrease in subject with MASLD? So the triglycerides are accumulating because there is no enough export of this TG within the VLDL. And so many studies still using tracer have looked at this. These are some of the most important. So the VLDL triglyceride production is actually increased with liver fat, not decrease. But you see here, it started to have like a sort of plateau. So it looks like the possibility to take out these triglycerides is limited. And of course, as the liver is very rich in fat, it reaches a plateau. But it's also interesting to look at the composition of VLDL that in both normal and NAFLD subject, the VLDL systemic was about the same, but the non-systemic fatty acid was high. But also it's interesting to look at that also in this study were found an increase in with lipolysis in this subject that have the similar body weight. The contribution of triglyceride-free fatty acid in fat and fasted state is very different. Of course, you see here the different time of the day, the fat and the fasted. You see here the contribution of free fatty acids is going down during fat state because of the insulin released. And so the contribution of the diet is increasing, but the contribution of DNL is about the same. When you look at the source of composition during the fat and the fast state to the VLDL and triglyceride-rich lipoprotein, you find that the diet is making a minor contribution. If you look here in this box plot that is summarizing these results, the great majority of triglycerides is coming from peripheral free fatty acids, so from the adipose tissue, about 26% from the DNL, and only 15% of the total VLDL is from the diet. So you look here how important is the adipose tissue on the composition and also on the source of the triglycerides in the liver and also exported as VLDL. Now, I want to switch to this new data that we have analyzed, and here is a review that we wrote last year, and also I will show you the paper that we just published. MASLD is not only about lipid metabolism, because glucose metabolism is also very important. And we know, as I told you before, that there is a link between MASLD and type 2 diabetes. There is an odd ratio of 2.2 for risk of developing diabetes in subject with MASLD. And so the glucose metabolism and the hyperglycemia is something we needed to study to look why these subjects have an increased risk. So a previous study on hepatic glucose, measure hepatic glucose production, but they were investigating mainly steatosis, and so they never looked in detail if there was a relationship with MASH and also severity of MASH, so MASH with advanced fibrosis. And this is what we have done in the other tracer. And it's important also to look because the excess lipolysis that we have shown previously with the adipose tissue insulin resistance impairs glucose metabolism and insulin signaling. And it has been shown that the lipid infusion has a major effect on the insulin receptors in the muscles. So excess lipids is actually impairing the glucose metabolism in the periphery and contributes to peripheral insulin resistance, but also hepatic insulin resistance. So we measure several fluxes, not only lipolysis, but also glucose production in the liver, and also the glucose clearance in the muscle. And the idea was to investigate how hepatic glucose metabolism was varying in the tradition from isolated steatosis to MASH, the impact of adipose tissue lipolysis on glucose metabolism, and if the stage of fibrosis has an impact on the glucose metabolism in the liver. But to do this, we used two cohorts. The first one was a relatively small cohort that was studied with the infusion of these tracers. They all had the liver biopsy. The first cohort didn't have diabetes, and we measure glucose production. That is called HGP, hepatic glucose production, hepatic insulin resistance, and adipose tissue lipolysis. The second one was also with liver biopsy, was a much larger cohort with liver biopsy transcriptomics, were with or without diabetes, and for this we measured the glucose metabolism intrahepatic using genome scale mathematical models. So the results were quite interesting. First, we look at hepatic glucose production, and despite the fact that they didn't have diabetes, we show an increase in this glucose production, even they don't have diabetes, and was an increase as a subject progressed from muscle to MASH with a very low fibrosis, intermediate fibrosis F2, or severe fibrosis F3 or 4. And you look here, the glucose as expected was not significant, though the last group 3 or 4 had tended to have a higher fasting glucose, and also the insulin instead were increased as a subject went from muscle to MASH 3 or 4. So we look, and there was a strong relationship between the adipose tissue insulin resistance and hepatic glucose production. And you see here the adipose tissue insulin resistance was increased with the fibrosis, as also in the previous studies. But also the similar trend we look at also with the HOMA-IR, that is another index. This is based on glucose, so it's glucose times insulin, so it's more related to hepatic insulin resistance. So the increase in hepatic glucose production was due in part to adipose tissue and in part to hepatic insulin resistance. So as I said, we use these genome scale mathematical models to investigate the intracellular fluxes, because these models that are quite new can be used to based on the transcriptomics. So this is the TCA cycle, this is glycolysis gluconeogenesis pathway, are important because based on these models that has been previously published and adapted to each individual that have the liver transcriptomics, it's possible to look at the fluxes. Of course, these were never been validated for the glucose metabolism, so we validated here versus the tracers. That is the gold standard. So we found that this model were very good to measure glucose metabolism, and indeed when we estimated hepatic glucose fluxes, we found still an increase in, although mild, in subject without diabetes, and an increase instead with the subject with diabetes. As expected, the glucose production was higher in diabetes compared to non-diabetic, but the trend with MASH fibrosis and fibrosis was there also in the transcriptomic group. So when we look at the hepatic insulin resistance, that is measured by looking at the relationship between the insulin and glucose production. So the higher insulin, you are supposed to have lower glucose production, so their products is the index of hepatic insulin resistance. You see here that in both the cohort, there is a trend to increase as a subject go from muscle, simple muscle, to MASH with fibrosis 0, 1, fibrosis 2, or fibrosis 3, 4. And it was interesting because we look at the expression of insulin signaling gene as RS1, RS2, and AK2, and we found not many differences in this subject. These light blue are the non-diabetic, and the green instead are the one with the diabetes. So with the higher fibrosis, there was a tendency in decrease on RS1, no change in RS2, and a slow decrease in AK2. And these genes were not used for the model, so it is quite interesting to look at that. Only in part the insulin resistance is due to the defects in RS1. So we also look if this was related also to other factors of histological factors like steatosis, fibrosis, or activity score, that is the ballooning plus inflammation. You see here that hepatic glucose production is increased significantly with fibrosis and activity score inflammation, while there was no significance with an increase in steatosis, although it seems here that there was a trend, and this was confirmed also in the transcriptomic cohort. Again, no change in steatosis, and although the LGP, as expected, was higher in the diabetic, that are the top circle, and again, fibrosis was associated with increase in hepatic glucose flux and also the inflammation. Now, we look with these models also intracellular fluxes. So it's quite interesting to have these models because it's possible to measure the intracellular fluxes that we usually can measure it with also deuterated water, but requires a lot of work and of course a lot of subjects. So previously, by using the deuterated water, we have shown that the gluconeogenesis is increased in subject with the diabetes, and so you see here fasting glucose, so hyperglycemia, and so the severity of diabetes is in the x-axis, and instead here we measure the gluconeogenesis and glycogenolysis. That is increased gluconeogenesis in obesity compared to the non-obese diabetic. And the liver insulin resistance also it's increased with obesity. And previously, we have shown that gluconeogenesis is associated with increased free fatty acids and with increased visceral adipose tissue, but is not associated with steatosis, with the percent of fat. So since gluconeogenesis main contributors to hepatic glucose production, you see here that the two components of hepatic glucose production, gluconeogenesis and glycogenolysis, the main impact is done by the gluconeogenesis. So considering that, we went to look with the genome scale what was the contribution of gluconeogenesis. And you can see here that as a subject become diabetic, there is an increase in gluconeogenesis, and there is also an increase of gluconeogenesis with fibrosis. So subject without fibrosis, without diabetes are this one, but there is a trend as subject go to more severe MASH, and of course, as they become diabetic. And these are the metabolic fluxes, all these fluxes that are considered here as an R. You can find it here, and to look that a subject with F2, F4, and type 2 diabetes have the highest flux through the entire pathway, glucogenic pathway. Also, the high glucogenic, we confirm here that it is the main driver of hepatic glucose production. And so we can summarize this part by showing that there is an increase of also hepatic glucose fluxes as subject progress from muscle to MASH with advanced fibrosis. This is driven mainly by fibrosis and inflammation, while steatosis is not making a major role. And we explain this with the increased substrate availability, mainly coming from the efflux from the adipose tissue, glycerol, and free fatty acids. And of course, is strongly associated with insulin resistance. So to summarize and finish my presentation, so there is a major importance of the role of the adipose tissue and insulin resistance to the development of the MASLD and also to the progression, not only through the excess substrate lipolysis, glycerol, free fatty acid to the liver, but also this one, as we saw, that are also accumulated in other organs, increasing insulin resistance. Insulin resistance is driving the excess requirement of pancreas, and insulin is also driving collagen and formation inflammation in the liver. And I like to thank my entire group. Most of these studies have been carried out by Fabrizia Carli and Silvia Sabatini, but of course there are all the other PhD student and the postdoc that are part of my group. And I'd like to thank you for your attention.